Name of the experiment: observation of visking tube permeability by using glucose and starch solution. Here I have taken two solutions, one is glucose and another one is starch solution, and these two solutions are mixed in a test tube. After that, this solution pass inside the visking tube. On the right side here is a diagram of digestive system, and we know that one part of digestive system is small intestine, and this small intestine, the features of a small intestine, it is very long, almost 8 meters. Its inner wall contain villi, which surface area for absorbing soluble food from a small intestine to blood. Villi surrounded by microvilli, which add extra surface area. It has one cell thick wall. It contains blood capillaries. So what we can do, at first we have taken these two solution in the visking tube, then place this visking tube inside distilled water in a beaker. After 5 minutes we will test the water in the beaker using iodine solution and benedict solution. After 5 minutes I am taking this solution in two test tube, and in one test tube I will add iodine solution and see the result. So no color change using iodine solution, and another test tube I will add benedict solution. This is the benedict solution, and place this solution in a water bath. Okay, now this is the iodine solution and this one is benedict solution. At first I am placing this visking tube with solution inside water, then we will wait at least 5 minutes. Here color change from blue to brick red, that indicates glucose is present here. Movement of water molecules from higher water potential to lower water potential through semi-permeable membrane. That's why that is the osmosis process happened in this experiment.